Um, so in order to do all of these tests that we're going to talk about, we have to generate antibodies. And there's a way to make antibodies. People do this commercially for all kinds of reasons. This term monoclonal antibody basically means that all of the antibodies are produced by one cell, and they produce all the same. Typically, this is done using mouse cells. At some point, you may run across the term polyclonal antibody. That just means that it was probably made in rabbits, and the way that you make them in rabbits, you end up producing multiple different kinds of antibodies. So we make these using what we call a hybridoma, which is an artificial cell that's created by fusing a normal antibody-producing B cell to an immortal cancerous B cell. So here's our B cell. Okay, this is a normal one that has whatever antibodies on it. This is what's the exact term that they use? Myeloma. So what is one of the hallmarks of cancer cells? It's distinct from body cells. Say again? Yes, abnormal growth. And what that really means is that they can go through cell division basically forever. Is this true of our body cells usually? No. Why would we have a limit on the number of times a body cell can divide under normal circumstances? Why would we have sort of a clock on that?
To try to prevent mutations.
Right. Every time you divide, you introduce at some low rate mutations, and it's risky to do that. So cells have actually, in our body, they have a natural lifetime. They'll die after so many divisions. But cancer cells have gotten around that. They, of course, don't really care if they accumulate mutations. That's to their advantage for the most part. So we take what we call an immortal. It's a B cell, so it's going to be similar enough that it's going to fuse to that cell. But it's changed its cell cycle regulation so it can divide forever. And we take a regular B cell that makes antibodies, and we fuse them together in order to make a monoclonal antibody, a hybridoma, something that's going to have sort of properties of both cells. It's going to have this specific kind of antibody that this guy makes, but it's going to have the ability to live forever. And if we're doing this for commercial purposes, why is that important? Why do we need to live forever? We need to be churning out antibodies. We want to be able to go back to the source and be like, okay, I ran out of that. I sold it to these 10 people. Now I need more. So this is how it's done. Here's our little mouse. You inject the mouse with antigen. So what are you putting into the mouse? What is that antigen you're putting in? It could be anything. It's anything you want to generate antibodies against. Okay, and I'm just going to say that for the purposes of, say, making vaccine, you probably are thinking, oh, it could be a viral particle. Maybe it's a whole viral particle. Maybe it's a heat kill virus. It's some kind of thing that you would vaccinate someone with. Maybe it's a subunit. Maybe it's DNA. But it could be anything. And in fact, people do this all the time to make antibodies against many things, not just for the purposes of looking at disease. In research in the field that I work in, we do this all the time because we want to look at where some protein X is inside the cell. It'd be really nice to have an antibody against that. So we inject whatever it is that we're trying to make an antibody against into that mouse. We let the mouse do its normal thing, right? We let the mouse's B cells, now remember, think about this, the mouse has B cells, generated the same way that ours are, which is to say that there's lots of random specificity. There's a huge, and so if some of those B cells have specificity against that antigen that we put in, that particular B cell is going to be selected for, right, and proliferate. We take out the spleen. It just happens to be one of the organs where you'll find a lot of immune cells, and some of those immune cells are going to be B cells that have specificity against that antigen. Now, it's important that the mouse has an immune response against the antigen because what is one of the first things that happens when B cell encounters the antigen? We replicate it. We make a whole bunch of them. So we're enriching the pool of most of those B cells are just like represented one time or a small number of times. That's whatever that antibody is, that specific antibody on that particular B cell is very rare until it meets the antigen. Then it becomes expanded, clonally expanded, right? So then we're taking the spleen out of this mouse that has seen this antigen, so it should have a lot more of that particular kind of B cell with that particular kind of antibody if it had an antibody that would recognize the antigen in the first place. So is there a risk that this whole thing could fail? What if the mouse doesn't have any B cell with any randomly generated specificity against this antigen? It's just really unlikely. So we take the spleen and we isolate out the B cells and we add in these other kinds of B cells that no longer make antibody. Okay? So in the process of becoming cancerous, they forgot what they're supposed to do, but they have the ability to live forever. Those two cells mix and some of them fuse into hybrid cells. We put them one each into a little well and we give them some food and they grow up. So this one hybrid cell here, all of these cells are related to this one cell in what way? They're identical. They were produced by mitosis. So we have a whole bunch of cells. Now, what else can we say? If there is an antibody type on this cell, what antibodies are being made by these cells? The same ones. All of these cells have just one antibody, right? What about the guys in this well next door? They are going to be made from a different cell. So it might be the case. I mean, there's a lot of possibilities. Let's say what we injected in was this protein that had a couple of different possible epitopes, right? And so B cell A has this specificity. So it's specific for this triangle. Sorry that my picture is so terrible. Then we have B cell B over here that has specificity for circular things, right? Both of these guys would be enriched in the spleen, and this guy A might be here, get bound to this epitope. And next door we might have a guy binding the same antigen but a different part of it, right? Or maybe they bind the same part, just with slightly different affinity. There's something different about them. They're unlikely to be identical because they're different cells and each cell has a slightly different specificity. But they might be binding to the same antigen or different parts of the same antigen. Most likely in these wells there's going to be some difference, however subtle, between the antibodies that are produced. And so one of these or more of these are going to be specific to the antigen. Can we also get stuff that doesn't bind this at all? Yeah. Right? Because there's lots of B cells in the spleen, not just the ones that are made when we injected the antigen. So each one of these guys, each one of these wells produces a specific type of monoclonal antibody, one kind. And then we can just do a bunch of tests with those and find the one that works the best. And then because they're immortal, we can just keep going back and make more.
Are there charges associated with binding, like negative and positive charge?
You're getting at what is the chemical or biochemical specificity, what is the basis of that? And yes, it is a lot of things. So charge could be involved. For instance, if what you're recognizing is protein, proteins are made of amino acids. Some amino acid side chains are positively charged, some are negatively charged, some are neutral, some are huge and lumpy. So it's shape, it's charge, it's hydrogen bonding, it's all of the things that cause different molecules to bind together non-covalently to recognize each other. Okay, so why do we do this? Antibodies allow us to detect things. We can do research with them. Also, remember what antibodies do in the body, like why we make them. They're effective against toxins and against microbes, right? In a number of ways. So when we're talking about what the reasons our bodies make antibodies, what are they doing? How are they effective against toxins? They can neutralize them, right? They can bind them, and then whatever that toxin was doing, because it's bound by the antibody, it might not be able to do that anymore. Some of the toxins have the ability to make holes in things, right? But if you have an antibody stuck on the side of it, it might have no ability to do that anymore. Also, because it's bound to antibody, that's a flag for your immune system to clear it. Okay? Same with microbes. In particular, things like virus and some bacteria, what is on their surface that's dangerous to us? How do viruses get inside? They use attachment proteins. Now, if you have an antibody that's stuck to the outside of a virus, it's going to have a hard time getting into your body. Also, you're going to opsonize it and flag it for destruction. So just some examples of commercially made antibodies that have various different kinds of effects. So this is a case where what we're doing is we're creating the antibody commercially, and then we're administering the whole antibody to the person who's sick, okay, for various reasons. So this one, Miromonab CD3, is effective against, it's basically, it helps people who have gone through organ transplant not to reject the new organ. So CD3, you may not have heard of, but you've heard of CD. What is CD? Where is that found? What cell type is that on? T cells. Now, are CD4 and CD8 the only CDs in the T cell world or not? There's tons of them. CD3 happens to be on the surface of T cells that are part of an autoimmune response. So they're particularly, CD3 is particularly dangerous if you just received an organ, because that organ will be perceived as foreign and you'll have an autoimmune response against it, partly mediated by CD3 on T cells. So if we introduce an antibody that binds to CD3, what possibly are we doing here? We are we're actually sort of reducing the effectiveness of the immune response. We're coating that CD3 on T cells and preventing it from recognizing this organ as foreign. Obviously, in order to have that kind of target selectivity, we had to develop a lot of knowledge about what's happening in an autoimmune response, what specific receptors on what specific cell types, in order to develop this kind of very directed therapeutic. Anybody with somebody in their family or friend with Crohn's or colitis? What is that disease? What happens? Yeah, you get inflammation, basically ulceration of the GI tract. Depending on where it is and how extensive it is, then it's diagnosed slightly differently. Crohn's is throughout the intestine. Colitis is more in the large intestine. One of the effectors of this disease, it turns out, is tumor necrosis factor. It's a cytokine, so it's one of these secreted proteins that talks to cells. And what it does is it causes cells to die. It tells your body cells to commit suicide. You go through this process we call apoptosis. Now, this is going to be a useful thing to do if, for instance, that cell is infected with a virus or a bacterium or something is bad about it and you have to get rid of it. But if we're just telling cells to die all the time willy-nilly just because, this gastrointestinal tract is upset, then we're going to have large amounts of tissue dying. And if we could turn this signal down somehow, that would really help somebody with this condition where their TNF is up too high. And so if infliximab is actually an antibody that binds to tumor necrosis factor and prevents it from working. We have some other antibodies that work for lymphomas. Lymphomas are, now that you know what lymphocytes are a cancer of lymphocytes with this particular B cell with a CD20. So CDs are not just on T cells, they're also on B cells. B cells with CD20 overproliferate, has various bad consequences. We don't need to get into that, but CD20 is targeted by this antibody. And so we can start to get rid of those overproliferating B cells. Anyone heard of Herceptin? Anyone in your family who has had breast cancer? Any friends with breast cancer? So not all forms of breast cancer are helped by this therapeutic, but it was really revolutionary because it's unlike a lot of breast cancers historically had to be treated with pretty aggressive and general chemotherapeutics. So things that, yes, they kill the cancer, but unfortunately have a lot of side effects. Herceptin was so great because it's an antibody against a receptor on breast cells, and so this is our breast cell. Here's the receptor. And what this receptor does is it talks to the cell and tells it to grow and divide. Normally, this receptor, which is called HER2, it only works sometimes. Obviously, our body cells, are they dividing all the time? I mean, I guess it kind of depends on which cell type. But are all of your body cells dividing willy-nilly all the time, all day long? Why would they initiate cell division? If you're a B cell, why do you initiate cell division? You've got to activate it. If you're a gut cell, let's say, why would you undergo cell division? Maybe one of your neighbors died and you need to replace him. Skin cells, same thing, right? Red blood cells are being regenerated because they don't live very long, right? So we do have cell division. It's very important, but we need to regulate it. In particular, in tissues like the breast or various kinds, especially in the brain, it's important to not divide and cause the organ to become misshapen. And it's also important that we don't divide too many times and introduce mutations. So this receptor, HER2, is on breast cells in some types of breast cancer. There's a mutation. And instead of waiting for a signal that would regulate when this grows and divides, we have a mutated version that just acts as if it's turned on all the time. So we developed this antibody against HER2, and we can actually prevent that receptor from having its function. This doesn't work in every case. It's complicated, but it is specific and can be very helpful. So antibodies, the point of this slide is not that you remember all this stuff, but that you understand that antibodies are specific, and because of that specificity, they can be deployed for a variety of things. There are a couple of terms you need to know, because as people have started to use this technology, they've developed ways to make it better. Because if you're using mouse antibodies, there's something about mouse antibodies that's sufficiently different from human antibodies that your body might not be happy to see them. So we can make chimeric monoclonal antibodies where we modify the mouse so that it produces an antibody. So normally, let's see, the Y-shaped antibody from a mouse. Okay, it looks like that. All of this is encoded by mouse DNA. We can change the mouse so that this region here is all made by human DNA. That's what we call a chimeric monoclonal antibody. These regions here, the ones that change, they're still mouse sequence. Humanized monoclonal antibodies are ones that are mostly human, except for the very tippy tips of the antibody, which again are where the antigen binds. And you can also have fully human antibodies, so the mouse just actually makes an antibody using a human DNA. Okay. So a number of ways that we can use antibodies. What we're trying to do in a lot of these diagnostic tests is we're trying to take some fluid, usually serum, and ask a question. Okay, we might want to know, is there antibody in this against something specific? Or we might be looking for some drug. For instance, if we're doing drug testing, we might be looking for some hormone. If we're testing someone's hormone levels, we're looking for something, okay? So what we can do if we're dealing with an antigen that's soluble. So again, this would be something like, many proteins are soluble. If you put them in water, they just sort of float around. What would be something that's not soluble? Something big, right? Like a whole bacterium is soluble? Yes or no? A whole bacterium is not soluble. Giant particle. viral particle isn't really soluble. So soluble antigens, like little proteins maybe, or sugars, they're dissolved in the serum. So we might be looking for those. We add some antibody. We add the thing we're asking a question about. And if this soluble thing binds to the antibody recognizes it, we see a precipitate. So normally we can't see with our eyes that there's something in there that we're interested in, but if we add the antibody, it forms this big clump and then we can see it. So that's what we call a precipitation reaction. If we're dealing with a non-soluble antigen, like a whole bacterium for instance, we can do an agglutination reaction. So here's our whole bacterium. Here's our, what kind of antibody is this? Of the 5 different kinds? IgM, right? And those are super good at agglutinating things, right? Because they have hands and they bind to multiple different things, and pretty soon you have a big clump. So that's an agglutination. Now, if our antigen is small and soluble, but we still want to do an agglutination reaction, we can put that antigen on latex beads and then put the beads in with the antibody. And this is what we call indirect, an indirect agglutination. So direct would be if it's a whole particle, and indirect is if we have to make it artificially into a particle. I'm going to skip that. Hemagglutination is a specific case of regular agglutination where we are clumping red blood cells. So why do we do this? It seems sort of crazy. This is because some viruses that we're interested in asking a question about, you might be interested in asking, has this person been exposed to this virus? And it's one of these viruses that binds to the surface of red blood cells and causes the red blood cells to crash out of solution, to agglutinate. So if we have, say, we're a patient, we've been exposed to that virus, so we have antibodies against that virus in our serum. One way to ask if that is the case, maybe as a doctor you suspect that patient has seen the virus, is if it's one of these viruses that normally agglutinates red blood cells, we add red blood cells and we add the virus. We have these parts, okay? Then we add this, which is the experimental part of the sample. This is the serum from the patient. And if this antibody recognizes this virus, it prevents it from attaching to the red blood cells, and we don't get an agglutination reaction. So this is called a viral hemagglutination inhibition reaction. It's a way to ask a question about what antibodies are in the patient's blood against some virus. We can also do a neutralization reaction, and so one way to think about this is it's based on the fact that this is what our bodies do normally, right? So toxins, some toxins, a lot of exotoxins, they damage cells, perhaps by making holes in them, for instance. And if we add antibodies against that toxin, we can prevent that from happening. The antibodies bind up the toxin. So let's say you were trying to ask a question about a patient's blood, right? Here's the serum, and we want to know, does this patient have antibodies against that toxin? We have the toxin, we have some cell that we know the toxin targets, and then we add the serum, and we see if the cell's damaged or not. And then we have an answer to our question. Another way of asking this question is using complement fixation. So this is kind of complicated and cool, but still complicated. So I'm going to step you through it, then we're going to go back and do it again. So basically, in the experimental sample, the person who has antibodies against this antigen, we add to that serum the antigen that we're interested in, plus complement proteins. And if the patient has antibodies against that antigen, the antibody is going to bind the antigen, and then we know that antibody-antigen interaction binds to some of the complement proteins. Via what pathway? In the body, when this happens, that's the start of which of the three ways of activating the complement? Classical. Okay? So if the patient has antibodies against whatever we're asking the question about, then this is going to happen. If the patient has never seen that antigen, has no antibodies against it, then the antigen and the complement go in, Nothing's been activated yet. It's important to recognize with this test that what this does is uses up the complement proteins. So next, we add two more things. We add some red blood cells and we add an antibody that binds to those red blood cells. That is going to be another antibody-antigen interaction, which normally would bind complement, but can't in this case. Why? We used it all up, right? We used it all up here. The patient's antibodies used up the complement by binding to its antigen. But over here, the patient who's never been exposed to whatever that antigen is and doesn't have the antibodies, when you add this red blood cell and this antibody to the red blood cell, the antibody antigen binds the complement proteins and causes one of the outcomes, which is to make holes in the red blood cell, causes it to undergo hemolysis. And so this is a way of asking, because we have these pieces, we have the red blood cells, we have this antibody, and we have the complement proteins, we have all those pieces. The only part we're asking a question about is this part. Are there antibodies in here or not? And if there are not antibodies, then what's the result? Hemolysis. Okay, so the complement is not fixed. Here the complement is fixed, it's used up, so we don't get any outcome. Same exact idea is applied to a situation where you want to ask if a particular organism, let's say, that is antigenic. This is streptococci, for instance, from a patient's throat. And we have this reagent, which is an antibody that we know is specific to this antigen, and it's already labeled with fluorescent dye. So we just add them together. We take our sample that we think has streptococci in it, we take these fluorescent antibodies that we know will react if there is anything there, and we just look to see if we get a signal. That's a direct fluorescent antibody test. We can also do an indirect fluorescent antibody test. So for instance, here is a parasite. We have, in this case, what we're asking for, so just to make it clear, because this is confusing and it's terminology that you'll just have to memorize however you do it. In this fluorescent antibody technique, and in most of these techniques, when we say direct, what we mean is we're directly asking, is the antigen there? In an indirect test, we're asking, is the antibody against the antigen there? So here is some parasite. There is some serum from a patient that we think's been exposed, so should have antibodies to that parasite. We mix them together, and if in fact we're right, then the parasite's going to be bound by antibodies. Then we add a secondary antibody that's going to recognize this antibody, and it's easy to generate these because this stem part here is antigenic. And we can create antibodies that we know will bind the stem no matter what's on the end of this antibody. So we get these secondary antibodies and we bind it to the first antibody. So in an indirect fluorescent antibody test, we're looking for antibodies against the antigen. In a direct test, we're looking for the antigen itself. Any questions about this so far? All of this is based on antibody-antigen interactions. The entire chapter is basically one concept. Say again? Unfortunately, what you do need to know is, you need to be able to define a direct fluorescent antibody test versus an indirect one. You'll need to be able to understand how the complement fixation test works, right? How precipitation, what's the difference between a precipitation test, which is against a soluble antigen, versus an agglutination reaction, which is against a particulate antigen. Okay, so basic, simple, definitional, memorize it, however you get it into your head, but it's all based on this specificity that antibodies have for their targets. I'm most likely going to test you on either the complement or ELISA if you're trying to decide which of those things in the textbook to focus on. Okay, just going to give you that heads up. Complement fixation test or the ELISA, or both, is what I'm feeling particularly. So ELISA, has anyone heard of this term? ELISA. Worked in the lab and done one of these. It stands for Enzyme Linked Immunosorbent Assay. ELISA, of course, is way more fun to say. basically in this case, what we have is an enzyme is linked to an antibody. So, again, it's going to rely on antibody binding to antigen. So here's some antibody with some specificity on the end. And over here is some enzyme. Usually that enzyme is peroxidase. And there's lots of peroxidases. They have their enzymes that are specific to targets. Usually what they do is they choose a peroxidase that has some substrate that when the peroxidase acts on it, makes another product that has a color. So we have our enzyme linked to antibody. Enzyme linked to antibody. The enzyme is going to, when it's working, it's going to produce some colored product. In a direct ELISA, just like in a direct fluorescent antibody test, a direct ELISA is looking for the antigen. Okay, so what we have is we have our little well in a dish. We have some like surface, and we coat the surface with an antibody that's specific to what we're looking for. So, for instance, what might we be looking for? Maybe we're looking for HIV, right? Does this person have HIV particles in their blood? Or do they have some particular bacteria in their blood? So we're going to take an antibody that would normally bind to that, okay? And there could be tons of antibodies that would react to the viral particle just depending on which antigen or which epitope it reacts to, right? We'll take one of those antibodies, we're going to stick it to this little well. This is a little cup basically with some fluid in it. Then we add the sample. If the sample has the whatever it is in it that we're looking for, it's going to bind to the antibody. Next, we add our ELISA, our enzyme-linked antibody, right? That is this guy linked to the enzyme. Also, same specificity. So we have the one that's fixed to the well, and then we have the same exact antibody that now has on it an enzyme. That's all stuck to each other, and then we add the substrate, X, and if this is around, it's going to cut the substrate and produce a color.
Well, I think let's stick to step 3. I mean, why do you have to have that first antibody there if you're going to add the...
That's a great question. Does anyone want to throw out an idea why you have to have the first antibody stuck to the little fluid cup? So it really is a matter of just understanding what's actually happening. It's fluid, right? So we stick this antibody like with super glue, not really, but basically with biological super glue. We stick this antibody down, and then we add our sample, and then we wash the crap out of it. Okay? So the only thing that's there's going to be a lot of stuff in that person's sample. There could be lots and lots of things. There could be a ton of other antigens, things we're not at all interested in, and just gunk. And we're going to add the sample, allow this antigen to bind, and all the other stuff that might be around is going to be washed out. And the only reason that this stays behind is because it's binding something that's stuck to the plastic well. Okay? It's a great question. So then we add this reagent, and again, we allow that binding to happen, and it's strong enough to withstand the washing that we're going to do now to get rid of other stuff. And then we add our substrate that turns a color if that's around. If we didn't do that, okay, if we went straight to step 3 and we just added the ELISA, right, there would be no way to tell if something was there or not. We add this in and then we add this without doing any washes, and immediately we get a color either way, right? Does that make sense? Yeah. Okay. So we have to have some way of washing the well out, basically, and that's why we stick it down. So it's a direct ELISA because what are we looking for in a direct ELISA or a direct fluorescent antibody test? What are we looking for? What are we asking a question about? The antigen. Is that antigenic thing in the blood? Is the microbe particle, is the viral particle there, is the toxin there, whatever it is you're looking for, is the hormone there, is the drug there, whatever it is that this antibody can be made specific to. And by the way, where did we most likely get an endless supply of this particular antibody? By doing that monoclonal antibody technology that we just talked about. An indirect ELISA. So in an indirect ELISA, just like in an indirect fluorescent antibody test, what are we looking for now? We are looking for whether or not the patient was exposed to the antigen and made antibodies. So it's indirect, right? We're not asking about the antigen. We're asking, did this person ever see the antigen and make their own antibodies? So what are we going to stick down to that well? The antigen. So then we add the patient's serum, and if it has antibodies in it, what happens? Antibody sticks to the antigen. Okay, now we're adding our ELISA, our enzyme-linked antibody, which is going to have its specificity is going to be to this, the flag part of the Y of the antibody. So it's specific to this antibody, not to the antigen. Grabs onto that antibody, and again, you're washing everything out. So anything, if this didn't happen, let's suppose that we stuck this down, the patient didn't have antibodies, so that didn't happen, and then we add our ELISA, what's going to happen? It all gets washed away, right? There's nothing to bind onto. So we add our color precipitate or our substrate that turns a color if peroxidase is around, and we get a color. The color tells us what? What is this positive result telling us? There was antibody, right? In a direct ELISA, what is this color telling us? The antigen's in there. Okay, so here, does anybody recognize what this is? Pregnancy test. Did you know that it was an ELISA? Did you know it was based on antibodies binding antigens? Well, I know that you've always wondered, what am I doing right now exactly? In so many ways, what am I doing right now? By the way, what was the result for you? Okay, so what we have is, it's a piece of paper to which is fixed our monoclonal antibody, and it's already, and we're looking for whether or not this person has antibodies against a hormone that's made during pregnancy. So on this strip we have the antibody and the hormone, and we're asking if in the urine we have these antibodies. So that would be, if the question we're asking is this around, that would be indirect. If the question, if we're doing this test and asking, is this hormone around, then that's direct, right? Does that sort of make sense? If the question you're asking is the hormone there, that's direct. If the question is the antibody there, that's indirect. So, in general, serological tests based on serum are direct if they are looking for the antigen, and indirect if they're looking for the antibody. And here are all the different kinds that we talked about. So you should kind of have a general idea of what the differences are. You should know direct versus indirect. You should know kind of what these different tests are, enough to be able to explain them briefly or recognize them on a matching question. Precipitation. Okay, the precipitation reaction, what's sort of unique about that? The antigen, we're asking a question about the antigen is soluble. An agglutination reaction, now our antigen is what? It's a particulate. It is not soluble. Hemagglutination, in this test, we're looking at the agglutination of red blood cells. And remember that the most common or useful application of that is because some viruses cause red blood cells to agglutinate. If we add someone's serum and they have been exposed to that virus and have antibodies, what's going to happen? The antibodies will bind the virus, preventing it from agglutinating the red blood cells. When we say neutralization, what are we talking about? What specifically are we saying the antibody is doing? It's binding to that toxin or that microbe and preventing it from attaching or preventing it from having some activity. Neutralizing it, inactivating it. fluorescent antibody technique is that one where we used antibodies linked to a fluorescent dye to either directly locate the antigen or indirectly locate antibodies against the antigen. Complement fixation. Remember that in complement fixation, much like in hemagglutination, we're using red blood cells, but instead of clumping them, we're looking for them exploding. Now, if we do the complement fixation and the result is that the red blood cells explode, what does that tell us? The patient had no antibodies, right? There was nothing to prevent that second step where you add the sheep red blood cells and the antibody to the sheep red blood cells. There's nothing to prevent that from activating the complement pathway. Finally, we get the ELISA, right, enzyme-linked immunosorbent assay, where we have an antibody linked to the enzyme peroxidase, and that's our indicator. Okay, so